Doctor, I'm a redwood and I think something is severely wrong with me.
Please, Miss Plant, tell me what is troubling you.
My other fellows call me Hobbit because I'm the shortest one among them and also have back pain.
Well, let's take a look at you and see what's going on inside. I think I can explain what seems to be the problem. But first, may I ask you some questions about your lifestyle?
Sure, Doc. Anything to cure my shortness.
Do you drink plenty of water?
Well, of course, Doc. I drink as much water as Mother Earth gives me. You know how mothers are.
What about your diet? Are you getting your daily vitamins and nutrients?
Yes, sir. Mothers make sure I get everything I need.
Well, my last question for you, Miss Plant. How much time do you spend outside in the sun?
Oh, Doctor, I don't like the sun. It makes me hot and dry. I prefer to stay inside where it's cool and dark.
Well, Miss Plant, I have good news and I have bad news. The good news is that you're not sick. The bad news is you're causing your own pain.
I don't understand.
Well, there's a natural hormone in plants called auxin that responds to sunlight. This little thing makes you tall, but also causes you to grow towards the sun.
Wait a second. So this auxin guy helps me grow up by injecting some drug in my back?
I think you need some further explanation.
Since ancient Greece, humans have shown great interest in the phenomenon currently known as phototropism. The famous Grecian philosopher Plato thought plants were sensitive to nature. Aristotle argued that, unlike animals, plants were completely passive and insensitive. During the Middle Ages, there were many poems and paintings about the phototrophic plants, but this interesting behavior of plants had not been researched by scientists until the late 17th century. Charles Darwin, a pioneer, was one of the first to research auxin and its effects on plants. His research explained the effects of light movement in grass coleopeptides, which is a protective sheath covering the emerging shoot in grasses. The results showed that when unidirectional light shines on the coleopeptile, it bends in the direction of the light. With the tip of the coleopeptile being covered with aluminum foil, no bending would occur towards the unidirectional light. Darwin's experiment suggested that the tip of the coleopeptile was the tissue responsible for perceiving the light and producing some signal which was transported to the lower part of the coleopeptile, where the physiological response of bending occurred. He then cut off the tip of the coleopeptile and exposed the rest of the coleopeptile to unidirectional light to see if curving occurred. Curvature did not occur, confirming the results of his first experiment. This experiment of the Arabidopsis plant was conducted in which both plants were under a 10-hour photo period of 22 days. The plant on the right was placed under cool white fluorescent light for 18 hours, while the plant on the left was kept in the darkness for 18 hours. As you can see, light plays a very important role in plant growth. It is known that phototropism was stimulated by light, but it was Julius von Wiesner who confirmed that phototropism is strongest toward blue-violet light. This provides the plant with means to optimize photosynthetic light.
In order to understand the mechanism of phototropism, first we need to look at the structure of coleoptile. The very tip of the coleoptile is necessary in light sensing. The middle portion of the coleoptile is the area where the shoot curvature occurs. The substance responsible for cell elongation is auxin. This hormone is formed in the cells at the tip of the shoot and is then passed from cell to cell. In this respect, auxins are responsible for cell elongation. Here is the stepwise mechanism. Once the light is sensed by the coleoptile, auxins will move away from the light and the concentration gradient of auxin develops on the shaded side of the plant. The high auxin concentration causes a decrease of the pH, making the cell wall less rigid. In addition, the acidic environment causes disruption of hydrogen bonds in the cellulose that makes up the cell wall. The decrease in cell wall strength causes cells to swell, exerting the mechanical pressure that drives phototropic movement. One of the most important discoveries by modern scientists in phototropism is the role of blue light. Scientists used coleoptile in their experiment and discovered that only the light with wavelength range from 330 to 550 nanometer could cause the curvature. People gradually realized that red light was not responsible for phototropism as they expected. In 1993, scientists found the first blue light receptor, cryptochrome 1, and later, in 1999, cryptochrome 2 and phototropin 1 and 2 were found. Finally, we are able to identify 4 kinds of photoreceptor of blue light. Modern study on auxin focus more on the transportation and distribution. Protein of PIN family are responsible for the directional mediation of auxin flux. Among all the protein, PIN1 is most important and dynamic for normal development.
I can't believe that humans know us better than we know ourselves. They know everything about us and we know so little about them.
Well, not everything. The question why plants grow towards the sun has been researched for hundreds of years, and there are still many puzzles left.
Are you kidding? Even I know that auxin causes phototropism.
You're a quick learner. Well, one of the most controversial questions about the photoreceptor is the UVB peak. Scientists debated a lot in the identification of an auxin transporter, but they still can't define an exact process for auxin redirection. Another puzzle is that after discovering the PHO2 receptor, people wonder what the role of the PHO1 and PHO2 photoreceptors are in this process.
Okay, cool. I don't understand what you're saying. The only thing I know is that I should go out, sit in the sun, and let most of my auxin go to my back. Thanks, Doc. Bye.
Bye, Miss Plant. I'm sure you'll be as tall as your fellows soon.